Already we have been studied the details about what do you mean by DNA and RNA. Moreover, what is the difference between DNA and RNA? Now we can easily recall what do you mean by DNA. You can see all types of cell. The cell is having its own controlling area. That controlling center of area is called nucleus. Inside of the nucleus, chromosomes are available. The chromosomes are always seen as a network in structure. We can call it as a chromosome when it becomes at the event of meiosis or mitosis, metaphase stage. The chromosomes are easily visible while we are observed under the microscope. Then we can call it as number of chromosome. What each and every species, it's having its own number of chromosome. So we can easily count the number of chromosome while we are studying under the microscope at the event of metaphase in any kind of cell division, mitosis or meiosis. The chromosomes number can be easily visible, and we can also easily visible the structure of chromosome. So inside of the chromosome there will be a multi-component that is called gene. That gene we can call it otherwise DNA. So deoxyribose nucleic acid, hence we can call it as DNA. So what now we can see about the importance of chemical composition of DNA. Already we studied the DNA is the nature of carrying the genetic information from one generation to another generation. So what is its main content of chemical composition of DNA? DNA is a complex molecule made up of monomer. Okay? Then we can call it as each and every monomer of DNA. Then we can call it as nucleotide. So what is the importance of nucleotide? What is its component? What is its important structure? And how do we call it in the name of nucleotide in various kinds, such as we can see one by one. Each nucleotide has three components. One is deoxyribose and then a nitrogen base. Third one is phosphoric acid. All of you see here. One is important of deoxyribose, second one is a nitrogen base, third one is phosphoric acid. The three will form a component, a composition, hence we can call it as nucleotide. Okay? So you must understand that deoxyribose means it is one of pentose sugar. Okay? Pentose sugar, we call it based on DNA, it's named as deoxyribose. Okay. Nitrogen base comes under two classification. One is purine, another one is pyrimidine. The purine have its own nitrogen base called adenine and guanine. Pyrimidine is another one kind of nitrogen. It's having its own base, cytosine, uracil and thymine, but here uracil is absent but thymine is present, cytosine and thymine. So in our DNA has purine base, adenine and guanine, pyrimidine base, cytosine and thymine. The deoxyribose is already we already notify it. There will be a pentose sugar. Okay, that pentose sugar linked with a base. A base means adenine, guanine, cytosine, thymine, whatever it is. But this pentose sugar linked with a base to form nucleoside. And third, phosphoric acid is not here. What do you mean by nucleoside? One pentose sugar and one nitrogen base. That's all. If it is the nucleoside linked with a phosphoric acid, then we can call it as nucleotide. So there is a two different between nucleoside and nucleotide. Nucleotide is only pentose sugar, deoxyribose, and then nitrogen base. It should be considered as a nucleotide, and one more phosphoric acid will linked. Hence we can call it as deoxyribonucleic acid. You please understand the importance of DNA chemical composition. Okay. Now we can see nitrogen base and then nucleoside, then third one is nucleotide. Student, nitrogen base adenine, then one base and then one sugar. If it is added means we can call it nucleoside. Afterwards adenine is considered about nucleoside, we can call it deoxyadenosine, and it will linked with phosphoric acid, hence we can call it as deoxyadenylic acid. Then we are comes under this guanine, then it will linked with sugar and base, we can call it as deoxyguanosine. If it is linked with phosphoric acid, then we can consider it is a nucleotide, call it as deoxyguanylic acid. Third one is cytosine. Deoxycytidine, when it is comes under nucleoside, afterwards it linked with the phosphoric acid, then we automatically we can call it as deoxycytidilic acid. And then thymine. Thymine, when it is considered about nucleoside means one sugar plus base, then we can call it deoxythymidine. When it is linked with deoxythymidine linked with the phosphoric acid means deoxythymidilic acid. These are all the way the chemical composition and its structure is completely known by us. Okay. What is the another more important? These nitrogen base and pentose sugar or pentose sugar, nitrogen base, and then phosphoric acid, we can call it as nucleotide. Many nucleotides are linked together with the help of phosphodiester link. Hence we can call it as polynucleotide. So many nucleotides are linked together to form in a double helix model of DNA. Hence we can call it as polynucleotide chain. Okay. These polynucleotide chain, when it is considered about DNA, these nitrogen base and sugar and phosphoric acid, both three are combined together to form nucleotide. There is no doubt at all. Okay. Suppose the double helix model, which will act as backbone of the DNA. There is one important question is always be asking in our NEET question, what is the backbone of DNA? Deoxyribose pentose sugar and phosphoric acid, both of them are act as backbone of the DNA, and adenine or guanine or cytosine or thymidine will emerge out from the link of DNA, and it will form a base pair in two linked base pair opposite in direction. Hence they are acting as both of them the DNA, and inside of the DNA These adenine and guanine can be linked with the help of hydrogen bond, glycosidic bond. Then we can see it furthermore in details.